Thank you very much for everyone joining the conference call today, where we will be discussing the results that we've just released on the phase 2 clinical trial of NLC V01 clinical trial of Tollovir, our dual action oral antiviral anti-cytokine compound for the treatment of hospitalized COVID-19 patients. I have with me today Dr. Dorit Arad, the Chief Technology Officer of NLC Pharma, inventor of the science and biology behind the Tollovir compound, as well as Dr. Jules Mitchell, our key clinical advisor, who's been instrumental in helping us complete the analysis of the data, as well as begin to prepare for the next steps in the Tollovir program. First and foremost, I want to share some context around the study, because this was quite an effort to bring this program in the face of a number of other oral antivirals that were in the marketplace or in development over the past year and a half. So as we started this work with Dr. Arad, and I'll allow her to comment on her work in a moment, she spent years, decades, really developing the science and the technology behind Tollovir, and getting us into a position where we could jointly move this forward. And we're very excited to be working with her as we move forward. What's clear is that this is unique, and it's unique not only because of the biology, but because it's addressing a key need that very few other compounds are. In the hospitalized setting, currently, really the leader in the space is remdesivir. And in this study, virtually all the patients were treated with remdesivir, both the treated group as well as the placebo group. So this standard of care did include remdesivir. And that's important because as we walk through the data, it's critical to understand that if there is any benefit with the signals that we saw, very positive signals that we saw in the study, this would be additive to what remdesivir could do alone. And as we move forward in the development, that is important to understand. Really, when you look at this hospitalized setting, there are very few treatment options, and there's been heroic work by physicians to figure out how to help manage these patients with competing symptoms. One of the big issues that we see is the negative feedback loop. So the virus taking over the body and triggering a hyperimmune response that is then further triggered by the virus, and being able to get out of that cycle is really critical if you're going to help treat these patients. So as we begin to move this forward really in a much more robust manner from here, we have to remain cognizant of the type of patients we're treating, but we also have to remain cognizant of the science of the Tollovir compound and how it could potentially be administered and used in concert with other therapies to change the course of individual patients' disease, and over time, hopefully, can have a significant impact in helping on a broader scale with many patients across many different locations across the world. So with that, the first thing I'd like to do is pass this over to Dr. Dorit Arad. What we will talk about a little bit here is the difference between the first cohort, the first 11 subjects, for which there was not significant data reported, because we really realized the power of the biology of the formulations we were working with as we started to prepare for commercialization of related products. And as we did that, we created new formulations. Dr. Arad created new formulations that give us the potential to have a pretty significant impact. So with that, Dr. Arad, I'd like to give you the opportunity to talk about how you came to the Tollovir version 1 from the work that you started in 2004, and now how you begin to move that towards the next phase with the second part of the study based upon the science that you've developed.
Okay, thank you, Gerald. So we have a composition now of some active molecules that we find in a natural route, that its extracts have been used for many years in various medical compositions and routes. We found these compositions that constitute Tollovir through a very thorough research that started, as Gerald said, more than 25 years ago. Actually, it is my lifetime research. I started my postdoc in UCSF when the late Professor Peter Coleman handed me a sequence of the 3C protease from the common cold virus, the rhinovirus, that was thought to be important for the reproduction of the common cold, of that virus. I worked on the structure, the unique mechanism of this enzyme, and mechanism-based drug design that was intended to inhibit this virus that is important for reproduction. Actually, the other group that was working then, many years ago, on the 3C protease was Agaron, that was bought some years later by Pfizer, and they also work with it ever since. At Tel Aviv University, a few years later, we found the unique molecule that showed nanomolar inhibition out of virtual screening of 300,000 molecules using computational chemistry and mechanism-based drug design computations. The molecule was found in the same route that we're using today. Then, at 2003, it turned out that the 3C of rhinovirus, or 3CL, same enzyme as in HRV, is also responsible for virus reproduction of the SARS, when the SARS epidemic emerged. We worked on several aspects of the SARS for several years, and 10 years later, the COVID came, and it became clear that the 3CL protease is the Achilles heel of the virus. I mean, stopping the 3CL protease would stop the virus. So we had the first version of our inhibitors, and because they were natural compounds, they were ready to go. Pfizer showed that the 3CL is a great target because now we see with its 3CL protease inhibitor Paxlovid, and we are very excited now to show the results of our double-blind clinical trial that shows that the Tollovir and its natural protease inhibitor shows you see the results. Actually showed a signal of good results, and Gerald will show you the results.
Thank you, Dorit. Thank you, Dorit. And I know the results are out there in the public domain, but we will just spend a little time here with what we have to go through them step by step. I think one of the key things as it relates to these results is actually the fact that they very closely mirror an earlier observational study that Dr. Arad conducted in 2020 that really guided the creation of the initial dosing regimen that was further tweaked as we began to use these products in different ways. And then as we began to move into the clinic in this double-blind placebo-controlled trial and ultimately improve upon what we have, we're now in a position where we can really drive things forward and make significant progress with regards to the development of these products. So with that, I'm pulling up the first, the second part of the study, which is really the focus of today's presentation, because this is where we see the benefit. So when we look at the top line results in the second cohort, we have n equals 11 on the Tollovir slide and n equals 9 in placebo. we really have to start to think about the difference and the meaningfulness of that difference within the context of hospitalization. The primary endpoint of the study was time to clinical improvement as measured by the National Early Warning Scale 2. As we see in the study here, there was a 2.7-day difference in the mean with Tollovir as compared to placebo. And again, this is in combination primarily with remdesivir and other standard of care compounds. This difference is quite meaningful. If you think about the potential risk of being in the hospital, especially in COVID wards, and further from a payer's perspective, reducing days in the hospital has a significant reduction in terms of reducing costs. So as we think about where we fit in the marketplace, we're not, at this stage, the focus is not the treatment of early patients. This is really the focus of treatment on later stage patients who have a significant burden, not only in the hospital system, because if they're in the hospital, that means they're not able to treat other patients potentially with COVID or other things in those hospitals. So there's a significant cost of having that patient in the hospital, as well as other procedures that are not being conducted that ultimately down the line will progress and could become more expensive and more dangerous for the patients. We also see on the median, obviously, there's a much bigger difference. And with these small n's, our statistician, Luis Rojas, has indicated it is very important not only to look at the mean differences, but also the median. And so when you look at these results, this is one of the key things that gives us confidence to be able to help patients improve. And that is obviously the top priority as we move forward. Going to the next key endpoint, days in the hospital. This is quite important because not only do you want a patient to improve, you want patients to improve and leave the hospital. As I mentioned, there are a lot of risks with being in these hospitalized COVID settings, and obviously very risky moving into the ICU setting. And so to be able to shave on a mean basis 7 days off hospitalization is extremely valuable for the patient, both for the patient's physical and mental well-being. We believe, although we don't have any data yet, but there's a lot of anecdotal data from other sources that being able to help patients improve faster will potentially, over time, reduce the risk of longer-term effects associated with COVID. And so, we are going to be following up with some of these patients as part of the protocol. And so there will be additional data over time that we'll hopefully be able to gather to really fulfill the picture around how this may benefit patients over the longer term. Now, when you talk about time to discharge from the hospital, we see there's not a significant, as much of a benefit there, but that's because the patients who passed away were not involved in that group. So this culled out the patients who passed, and obviously the patients who passed are the ones that really stayed the longest, some over 2 months in the study. And if you think about what it means to be able to get patients out of the hospital quickly, what that means is that it's much less likely that they'll have some other chronic affliction that keeps them in the hospital for that significantly long period of time, 4 weeks, 5 weeks, 6 weeks, 7 weeks, 8 weeks, which can really drain resources, both from a capital perspective and from a utilization of the hospital perspective. So being able to get those patients out is quite important. When you look at the benefit of deterioration to mechanical ventilation, so that's keeping people off of ventilators, there was a 24% lower likelihood that you would become a ventilated patient on the placebo, on the treatment arm versus the placebo. So there was a 33% chance in the placebo arm and only a 9% chance in the treatment arm. If you look at it on a relative perspective, obviously it's quite significant in terms of the reduction of patients who move to mechanical ventilation. Similarly, There were no deaths related to COVID-19 while on treatment of the Tollovir group versus 2 deaths. And again, this mirrors very closely the same data that we saw in the observational study. So we saw a reduction in time in the hospital in the observational study. We see actually a bigger magnitude here in the placebo-controlled study. We saw a significant reduction in patients deteriorating to mechanical ventilation in the observational study. That is, mirrored here as well. And then we also saw no deaths related to COVID-19 in the observational study, and we mirrored that here. In terms of total deaths, there were 33% of the patients in the placebo arm passed versus 9% in the treatment arm. The one patient who did die, quite unfortunate. We do see there's an asterisk, and so we'll note that. The patient actually had a stroke prior to receiving study treatment. The patient, while on stroke under mechanical ventilation, began to receive Tollovir treatment through the feeding tube and actually did achieve, was a responder in terms of the primary endpoint of time to clinical improvement. Unfortunately, this patient, the protocol called for 10 days of treatment, regardless of whether or not there was clinical improvement or he'd left the hospital. And so we could not continue to treat that patient. We obviously don't know what would have happened, but unfortunately, 9 days after the treatment stopped, that patient began to experience difficulties and expired. So one of the lessons that we think we're going to learn from this is going forward as we plan the next set of studies, we do intend to potentially treat for a longer period of time. And this trend is something similar that we've seen with other compounds working in the hospitalized setting. That 10-day treatment may not be long enough for some of these patients who have other issues. And so, we don't know what would have happened, but that is something that we will be considering as we build out the next study. And certainly, once I'm done here, I'll allow Jules to speak a little bit about the next steps for the program. In terms of the need for supplemental oxygen, there was this reduction of 16%, 8 out of the 9 patients on placebo arm versus 8 out of the 11 in the Tollovir arm. But interestingly, the time on supplemental oxygen is quite significant. So there were 2 days less. Instead of 5.8 on average, it was 3.8 on average days on supplemental oxygen, with the median being 3 days versus 7 days in the placebo group. So while these are small ends, this is a very good signal all around as it relates to the clinical benefit of Tollovir. Now we're moving on to the biomarker analysis, which is preliminary. There is still a significant biomarker work that is ongoing, and we will have additional data as it relates to other biomarkers. And we are working on making sure that we get that in place now. But looking at the data that we have, really the focus, it was challenging because of the variability to think about where a patient started and where a patient ended. On some levels, some of the data actually might look better if you did comparative. But I think the focus really is, patient comes into the hospital, regardless of where they start, the key is where they finish. And so if you can normalize these numbers, you give the patient a better opportunity to have a successful outcome. And so when we look at C-reactive protein, very similarly to what we saw in the observational study, there was a very significant reduction in the small ends, but overall, there was a reduction of 11 on the C-reactive protein, which represented an over 75% as compared with placebo. And when you look at the median, that is even stronger. So it's small ends, but it is quite significant and is certainly trending in the right direction. Similarly, with IL-6, which is a very strong indicator of poor clinical outcomes, we saw that the Tollovir arm was better than the treatment arm. And similar story on the D-dimer, although the number of ends in the placebo group is quite small, and we'll be doing some additional work to make sure we get all of the data as the next stage in the biomarker analysis alongside some of the other biomarkers, we see, a quite significant lower amount of D-dimer in the Tollovir group as compared to the placebo group. So, taken together, which is the way I think we need to look at the biomarkers, the Tollovir group appears to have a much stronger signal of efficacy, obviously, than the placebo group, which I'll remind you, the placebo group does include remdesivir as part of the standard of care. So with this data, the challenge, of course, that we had is that this is a smaller study. As we know, Todos is not, and NLC are not huge companies. And so we went after the most difficult to treat population of patients, that there was a significant amount of competition in terms of accessing site and recruiting, because Israel is obviously one of the leading places where these studies are done. Notwithstanding those challenges, and notwithstanding the small ends, we do see very strong signal across the board. And so this gives us great confidence that we're on the right path with Tollovir, and now we need to think about the next steps. In terms of how we came to the conclusion to stop the study, as we saw the safety signal with a fourth death in the study, that triggered an analysis. So, we had to look at that because there was a risk that it could be treatment related. And so that was the primary cause of pausing the study, to allow us to investigate and identify whether or not it was treatment related. And as we went through the preparations for the analysis, really we started to think about potential efficacy and what that would look like and what the parameters were for efficacy given the small number of ends. And it was really determined that if you had a 20% plus improvement, this was coming from our statistician, Dr. Luis Rojas, that in combination across multiple clinical and biomarker endpoints would represent success. And so that was the bar we set for ourselves when we blinded the study, and based upon all of the data, each of the parameters that we're looking at, we succeeded in achieving that objective. And so it was recommended that we stop the study because continuing to enroll and getting to 70 some odd patients would not have had the effect of allowing this to become a pivotal study. We still would have to go out and do another study. And in light of the changes over time for patients dealing with COVID-19 in the hospital from the time the study started, which was in late 2020, to now, early 2022, we obviously think we can do a better job in designing the study, putting together some of the learnings that the field has made over the last year, and now really put something together that is robust, that will be bulletproof in terms of being able to win over regulatory agencies worldwide. So with that, what we want to do from here is really start to think about the next steps. And as we think about the next steps, the key for us is really building upon the work of Dr. Arad with the observational study that was done in 2020, really helping solidify the dosing, the work that was done in terms of the material qualification. So now we have a very robust understanding of the 3-cell protease inhibition, the specific compounds that are responsible for that inhibition, how we create the mixture that is now the Tollovir mixture that has both very strong anti-cytokine and antiviral activity that we think can stop the negative feedback loop associated with late-stage COVID, COVID that results in hospitalization. And now we can really, in a diligent manner, start to think about the next steps. The plans obviously are to take the adult hospitalized study into a later stage phase 2/3 study, and then also look at the other areas where this dual mechanism could have a benefit and where the significant safety profile, so we had no safety signal concerns in the study, where the very significant safety profile of this compound, where, as Dr. Arad mentioned, This is known to be safe, and we do believe it is safe enough for pediatric patients to use. With rising hospitalization rates among children, you can start to think about this compound within that context. Looking beyond a hospitalized setting for adults and children, we need to look at preventing people from deteriorating to the hospital. And that's where Gilead really showed their benefit. It didn't appear that there was much benefit actually in the hospital with remdesivir, although it is used and it is the standard of care, because currently it is the best thing that we have. And we think that we could be better. But now Gilead is very focused on outside the hospital, because they are showing benefit there, which is obviously very good for patients. We also think that we can show benefit there with our dual mechanism. And we also see that Gilead is now trying to prepare an oral formulation of remdesivir, because it's not really feasible to treat large numbers of patients in an outpatient setting that are moderate COVID. It's already significantly challenging treating early stage patients with monoclonal antibodies, and that's been a huge effort from people all across the country, and that's unfortunately shutting down now due to some of the biology of those compounds as it relates to the currently circulating variants of concern. With that said, it still is a challenge delivering IV in the outpatient setting. And so they're moving to oral. We're in the oral space. We think that our compound is better, and we're hoping to prove that as we move to the next stages. And then obviously in a somewhat different setting, the post-COVID sequelae. What we're starting to hear both from physicians and in the literature is that patients with Omicron especially are getting reinfected, and that reinfection may be as a result of not actually fully clearing the virus the first time you have it. And if that is the case, and I'm sure it is the case for at least a subset of long COVID patients, having an oral antiviral that's taken over a period of time could tip the balance in favor of eliminating virus that is dwelling in certain places in the body. And so looking at that is something that we think from a chronic disease management perspective is likely going to become one of the biggest issues going forward as a result of the pandemic that we're going to have to deal with for years to come. And we think it's a very significant opportunity to help patients in that setting. So with that, those are kind of the corporate plans. And I'd like to turn it over to Dr. Mitchell for just a few moments to talk a little bit about his experience in working with products like Tollovir and how we might approach regulatory bodies to think about bringing this into the marketplace.
Okay. Thank you, Gerald, for a very comprehensive presentation. So I want to just confirm that that based on the data, and I'm a data guy, that there was a strong signal that there's potential for this compound. Just as a general background, I come out of basic research, worked at AIRS, which became Wyeth, and then 4 really good years at Pfizer. Still have a few friends there. And then we set up a CRO back in the early 90s, and responsible for many products approval, including evaluations of botanicals, one in burns and the other one for ulcerative colitis that came out of traditional Chinese medicine. And so the area here in the area of botanicals is very exciting. And traditionally, until relatively recently, I'm just saying a rough number, but half the products that medicinals came out of botanicals, many and mechanisms of action. And apparently plants have a good way of dealing with viruses also. The current plan is to approach regulators globally, different geographical areas that have different geographical needs, but primarily with their unmet medical needs, and discuss the development program. We expect it to be different in different areas. But I think there'll be a strong reaction to these kind of data, primarily because of the apparent safety of the product, and also that it's a natural product with low levels of concern raised safety. So I think that as we deal with the regulators, I think there's going to be different approaches depending on the geographical areas. Some of them may view this on emergency use authorization, even based on what we have now, and others may want additional data. But I think we'll come up with a comprehensive plan for both developing world, where I think there could be very strong unmet medical needs for this product, as well as in the so-called developed world, where a product like this, even on top of existing therapies, or even as an outpatient product, okay, may be easier to deal with than some of the more advanced products, and in terms of stability, in terms of ease of use, etc. But it'll all be about the data. Okay, and as I said earlier, I'm a data guy. So there's two groups we have to satisfy. One is the data, and we have that already. We're planning to do a follow-up study that could be used for regulatory approval, either as emergency use or as full approval, and as well as just exploring the different areas of the world where we can possibly even get this onto the market sooner. So we have to deal with both. Now that we have the data, I think now we have to approach the regulators, give them a comfort level at what we have, and find out their needs as to what they need to make this a commercial product. Thank you, Gerald.
All right, thank you, Jules. So in closing, yeah, I think it's quite clear. We're very pleased with the results. I think Dr. Arad obviously has to be commended for the tremendous work that she's done throughout the years to help bring these products to patients. And now, we are able to help patients, and that is very gratifying. Certainly, we hear from people that the benefit that they've had is quite significant. I know Dorit also, and Avram, who's there, although on mute, also has been helping many patients in Israel, and hopefully soon we'll be able to start helping patients in a more robust manner here in the United States. So our plans from a commercial perspective, as many of you know, we are a commercial stage company. We do in concert with NLC, and we are finalizing our transaction as we speak. We do have capabilities to manufacture and actually distribute product, get it to people, and help them now. And so we intend to continue to build upon that. We have been sourcing the materials to be able to manufacture at large scale, and we're going to continue to drive that message because governments we've seen have ordered kind of the outpatient oral antivirals, 5 or 10 million a clip, and even some of the larger companies like Pfizer can't deliver in a short period of time. We know that we can deliver millions of doses as needed, and so as we start to build up, and now that we have this placebo-controlled randomized data supporting our hypothesis, and as we start to build out the next steps of taking this into a more robust clinical development plan and potentially getting early emergency use authorizations in certain jurisdictions, we can deliver the product and begin to help patients now, and the revenue generated from that could have a significant benefit in terms of allowing us to take this over and really commercialize this on a grander scale, as we've seen with other companies in the vaccine space. With that said, we understand the challenges in bringing these products to market, and it's not only being able to manufacture and sell. And so we are certainly open to relationships that can allow us to bring these products to patients faster. And over time, we expect that there's going to be a lot of opportunity for us based upon this. So with that, I will turn it back over to Sequire. And I'd like to thank everyone on the call for their help. And I'm not sure, Dorit, if you'd like to say any last words.
Yes, first I would like to thank the team of the clinical trials because they really did amazing work in analyzing data and also in treating patients. And I would like to thank Todos for enabling this study and enabling this work, and Gerald especially. And I hope we can bring it to many people, to help many people.
Thank you. And I'd like to add, I want to thank obviously Dorit and Avram for the tremendous work that they've been doing, but also Dr. Mitchell, who really stepped in and got his hands dirty to help really guide this analysis and allow us to bring really solid, credible data that's not overstated, but really presents what we have, which is significant, and gives us the opportunity to take it to the next level in a robust manner so that we can bring it to patients.
And I would like also to thank my partner Avraham Marilus, who had a tremendous role in initially bringing it to people and making a great effort all the time to save people and to promote this molecule, the Tollovir. And of course, Jules Mitchell, as you said. Jules, thank you.
You're welcome. And I just want to make one brief comment that and this is an excellent team led by Gerald and Avraham, and committed, everyone committed to really studying this product properly and making sure that it's safe and effective, and if it does work well, to make it available to mankind. And so thank you.
Thank you.
All right, thank you. So this concludes the presentation, and I'd like to thank everyone for having participated. Thank you.